the elemental impurities are not always tested for the formulation, and the elemental impurity risk assessment is done for every pharmaceutical formulation. If in the risk assessment it is justified that the product doesn't contain the elemental impurities, or by the risk assessment if it is demonstrated that the elemental risk is low, then the elemental impurity testing is not required. That's why understanding about the elemental impurity risk assessment, abbreviated as EIRA, in the pharmaceutical industry is very important to understand. Welcome dear viewers to this video, and let's start with some introduction part. What is the objective of the EIRA? So the main objective is to confirm whether the elemental impurities might be present, or if there is a presence of elemental impurities, then these are present at how much level, and these practices are done as per the ICH guideline Q3D. As we know, there are multiple impurity types like organic impurities, inorganic impurities, volatile impurities, elemental impurities, and nitrosamines. Out of those, elemental impurities are evaluated in the pharmaceutical product. The objective of EIRA is to identify, to evaluate, and to control the toxic elemental impurities in the drug product, and thereby to ensure the patient safety by maintaining the exposure levels below the permitted daily exposure, that is PDE, as it is given or defined by the ICH Q3D guideline. So for elemental impurities, the limits are given in the PDE values or permitted daily exposure values. Another objective of risk assessment for elemental impurities is to provide a scientific and regulatory justification for impurity control. If through the risk assessment you find that there is no risk, then the testing for elemental impurity is not required. And considering the daily dose of the formulation, the elemental risk assessment is done. I have made some videos on elemental impurities, so I suggest you to go through those videos to understand this topic. For this video, I am considering the risk assessment part. For doing the risk assessment, we should understand what are the probable reasons for the presence of these impurities, or probable sources from where this impurity may come in the product. So the sources are raw materials, sources are the equipments, and the container closure systems. Understand that elemental impurities are not the degradant type of impurities, so this will not increase, and that's why these are not tested for the every product. These are not tested in the stability. Only in the initial part, if you do risk assessment, and if these are not exceeding the limits, then there itself you can justify that you will not test. So it is different from the other impurities. So sources may be drug substance, API, excipients, which may be from the natural source or mineral sources. Generally, the mineral sources, excipients like talc, may have the content of elemental impurities. Then water, whether the water is purified water or WFI, catalyst and reagents used in the synthesis of the drug substance and APIs. Then excipients may introduce some impurities. Manufacturing equipments, stainless steel, glass contact, and container closure system, which may be plastic, rubber, or glass. But generally, in the pharmaceutical setting industry, the manufacturing equipments does not leach, or generally as are considered as not uh, responsible for the elemental impurity as a source. Then Q3D, elemental impurity classification. So as per this guideline, there are 4 classes, class 1, class 2A, 2B and 3. So for class 1, toxicity is high. These are AS, CD, Hg and Pb. Class 2A elemental elements, generally these are toxic, cobalt, vanadium and nickel. Class 2B lower probability, and these are listed here. In the example you can find like selenium, ruthenium. Then class 3, these have the low toxicity, and these are considered for the oral formulations also. These are like barium, chromium, lithium, molybdenum and many others. Now risk assessment step 1. So once you start the risk assessment, first is the identification step. Prepare list of all potential elemental impurities as per ICH Q3D for oral formulations and get their PDE values. Consider the raw materials, processes and packaging materials. Use supplier certificates, literature and prior knowledge. Collect all the declarations for the elemental impurities for each of the material, whether it is API, excipient, coating material, capsule shell, and bases. Consider all. Consider packaging material also. Then risk assessment step 2, evaluation of the sources. Collect the information from the COS. Collect the information from the declarations. Equipment material composition, container closure, leachable data and published literature and the historical process knowledge. Then risk assessment step 3, estimation of the exposure. So what is the dose of your drug product? Then consider the formula. Exposure limit microgram per day, impurity concentration into max daily dose in gram per day. So impurity concentration will be microgram per gram and the max daily dose will be gram per day. For cadmium found at 0.5 ppm in the excipient, daily dose is 2 gram, so 1 microgram daily exposure will be there. Then step 4, comparison with the PDE and risk characterization. Compare the exposure as per the PDE values given in the ICH Q3D. Example we have taken for the cadmium. Cadmium is 5 microgram per day for oral route, 2 microgram per day for the parenteral. Risk levels, low risk means exposure is less than PD. Potential risk, exposure close to PD values, so monitoring needed. And high risk exposure exceeds the PD. So redesign the formulation, material substitution, you can change the source as well, or you can use that material at some lower level. Otherwise, you will have to do the testing for each and every batch. Now coming to the control strategy. So whatever the risk assessment you have done and risk found, based on that you will do the risk control strategy. Supplier qualification and monitoring, routine periodic testing for the high risk materials, equipment cleaning and validation, container closure system evaluation, change management for new materials or vendors, and then outcome and deliverables. So outcome will be assurance that the elemental impurities are within the safe limits. Deliverables are like list of elements considered. You will prepare a report and it will contain the formula of the formulation, process, container closure information, then list of the elements which are considered for the risk assessment, data sources and evaluation methods, then PD values, references for the risk assessment, daily exposure calculations, PD comparison table, risk ranking and the conclusion, and then you will show the control strategy. This is the information for the elemental risk assessment for the elemental impurities or risk assessment for the elemental impurities. In my experience, I have found that generally the presence of elemental impurities is not there, or generally it is not tested for the oral solids, as the commonly used excipients are there and this does not contain much higher amount of the elements, and that's why the risk become low. But if you are using some mineral excipients like talc, so sometime if daily dose is much more and if you are using talc at higher concentration, there are chances that in risk assessment you will find the PD values are going up, and that's why the testing is required. And if testing is required, it will be done at the initial stage only. So this is the information regarding the elemental impurity risk assessment. I suggest you to go through my other videos on the same topic. Thank you.